for decades, oncologists who treat this really rare brain-adjacent tumor have been like playing a high-stakes game using a rule book that was just completely wrong.
Yeah, totally wrong.
I mean, imagine this medical mystery located right at the roof of your nasal cavity. We're talking about a cancer so incredibly rare, it only affects roughly 0.4 people per million each year.
Right, it's virtually unheard of for most doctors.
Exactly. And it sits right at the threshold of the brain, kind of hiding in plain sight. And historically, it's carried a coin flip, 50% survival rate over 10 years. But today, we are looking at a map that didn't just bend the rules of how we understand this disease, it completely shattered them.
Yeah, because when a disease is that rare, gathering enough raw data to truly understand its mechanics is just a monumental challenge. I mean, you're usually working in absolute diagnostic muddy waters.
Muddy waters is a good way to put it.
Right. You're making these educated guesses based on tumors that just look somewhat similar under a microscope.
Which brings us to our mission for this deep dive. We're exploring olfactory neuroblastoma, or ONB for short.
ONB, yeah.
And our source material today is this really groundbreaking study from Nature Cancer. We're going to decode this highly complex tumor ecosystem, uncover a hidden cellular identity that is actively rewriting the medical textbooks, and explore how this single discovery is like paving the way for true precision chemotherapy.
It really is a massive shift.
Okay, let's unpack this. We're going from an intimidating scientific paper to a fascinating journey of cellular discovery. And the key that unlocked this entire mystery is this tool called single-cell RNA sequencing.
Right, and to truly grasp the magnitude of single-cell RNA sequencing, or cRNA-seq, we really have to look at the old standard first, which is bulk RNA sequencing.
Right.
Historically, if a surgeon removed a piece of a tumor, scientists would take that entire piece of tissue, grind it up, and read the genetic material as just one single average.
I always picture bulk sequencing like making a fruit smoothie.
Oh, that's a great way to think about it.
Right. Like if you take a fruit bowl containing strawberries, blueberries, bananas, and you throw it all into a blender, you get this blended mixture. You can taste the overall sweetness. You know there's fruit in there, but you've fundamentally destroyed the individual pieces.
Exactly.
You can't tell how many seeds a specific strawberry had, or if one single blueberry was uniquely sour. Single-cell RNA sequencing is the scientific equivalent of unblending that smoothie.
Unblending the smoothie, yeah. That physical separation is the vital first step. Instead of getting a blurred average readout of a tumor, researchers are isolating the fundamental unit of life, the individual cell.
Wow.
Right, they're reading the exact transcriptomic profile, which is basically the specific set of genetic instructions that are actively being executed by that exact cell at that exact moment.
It's just mind-blowing that we can do that now.
It really is. And for a cancer this rare, the scale of this study is just staggering. I mean, they took samples from 10 treatment-naive ONB patients alongside one healthy olfactory tissue sample and analyzed over 96,000 individual cells.
Wait, 96,000? That is a massive data set.
Massive.
But, I mean, that presents an immediate glaring problem,
Right.
doesn't it? When you pull a piece of tumor tissue out of a patient, you aren't just pulling out pure cancer.
Right, you're extracting an entire microscopic neighborhood.
Exactly. You've got immune cells, structural cells, blood vessels, and the rogue cancer cells, all just living mixed together. So if we want to study the enemy, we first have to separate the malignant cells from all those innocent bystanders.
And they accomplished that using a really cool biological phenomenon called copy number variation, or CNV.
CNV, okay, break that down for us.
So a normal healthy cell is incredibly strict about its DNA packaging, right? It has a standard, highly regulated set of chromosomes. Cancer cells, by contrast, are just genetically chaotic.
Chaotic how? Like mutated.
Yeah, they often possess abnormal polyploidic chunks of chromosomes, meaning entire vast sections of their DNA are duplicated, wildly mutated, or just deleted altogether.
So they're looking for the cells with this chaotic, hoarded, or shredded DNA. But wait, to spot the abnormal, you need to know what normal looks like in that specific patient's nose, right?
Spot on. So they established a baseline by isolating normal fibroblasts, which are a basic type of structural cell, from the healthy tissue sample.
Okay, that makes sense.
By holding the genetic profile of the tumor's epithelial cells up against those healthy, stable fibroblasts, their computational models could actually flag the wild chromosomal variations.
Oh, wow.
Yeah, so any cell showing those chaotic CNV patterns was tagged as a malignant cancer cell. They systematically filtered out the healthy neighborhood to isolate just the rogue elements.
Okay, let me make sure I'm tracking this. We took 96,000 cells, unblended the smoothie, and used chromosomal chaos as a measuring stick to isolate the pure cancer cells.
That's the exact process, yeah.
And this is where the old medical textbooks basically get thrown out the window. Because historically, oncologists treated ONB as a strict binary disease.
Right, the classic either or.
Yeah, you either had the basal subtype or you had the neural subtype. But wait, if they've been treating this as a simple coin A or coin B situation for decades, putting these cells under this kind of high-resolution microscope must have been a massive shock to the system. Did that whole binary theory just fall apart?
Oh, it completely collapsed. Yeah. The researchers applied a mathematical pattern-finding algorithm called non-negative matrix factorization, or NMF.
NMF, got it.
And instead of forcing the data into pre-existing human categories, this algorithm looks at millions of genetic data points and just finds the natural harmonies in the noise. And it proved that this tumor isn't a two-sided coin at all.
What did it find?
The algorithm actually revealed 5 distinct metagene modules driving this cancer.
5, so 5 distinct genetic neighborhoods operating inside the tumor.
Exactly. Now, some of these modules made sense based on the old binary system. Like Module 1 was highly enriched for olfactory neurodevelopment, which maps perfectly to the old neural subtype.
Okay.
And Module 2 was entirely dedicated to cell cycling, which maps to the aggressive basal subtype.
But what about the other 3?
Well, the algorithm spit out modules related to cilium movement, and crucially, modules heavily related to tumoral mesenchyme.
Hey, mesenchymal?
Yeah, those mesenchymal modules are really the pivot point of the entire study.
Oh, wow.
The algorithm was highlighting cells whose genetic programming was dedicated to organizing extracellular structures and responding to fibroblast growth factors. They were staring at a complex, interconnected society of cells that was vastly more sophisticated than a simple either-or scenario.
So just to do a quick recap here for you listening, because we're about 5 minutes in. ONB is this rare nasal cancer with a 50% 10-year survival rate. Single-cell RNA sequencing lets us look at individual cells, and CNV helps us spot the mutated cancer cells.
Right.
And the big takeaway so far, the old binary system is dead. Think about a time you assumed a problem only had 2 solutions. What happens when you zoom in and realize there's a third? Because that's exactly what the researchers found next.
Yeah, which brings us to the massive biological breakthrough of this paper, the NBM classification system.
The NBM system. So we're officially replacing the old binary with a new trinity: neural, basal, and mesenchymal.
Exactly.
Let's break down the unique personalities of these 3 subtypes, focusing on how they actually behave in the body. Let's start with the basal subtype.
Okay, the basal cells.
I pictured these as the fast dividers.
Yeah.
If you look at their active genetic instructions, they are just overwhelmed by cell cycling pathways, right?
Yeah, specifically genes like MKI67 and TOP2A.
Right. They are expressing genes whose sole biological function is to basically yell, divide, multiply, and spread.
It's all about rapid expansion.
And because their entire existence is focused on rapid replication, this subtype is highly aggressive. Clinically, it correlates with the most severe dedifferentiated tumor grades, which are known as Hyams grade 3 and 4.
That's right. And you have to contrast that with the neural subtype.
The brainy ones.
Yeah, the brainy ones. These are cells that still kind of remember where they came from. Their active genetic programming is desperately trying to build neural pathways and facilitate olfactory transduction, which is the actual mechanism of smelling.
That is so weird. Even though they're cancer, they're trying to smell.
Essentially, yes.
Yeah.
Even though they are mutated cancer cells, they're expressing genes like CHGB and NeuroD1, trying to wire themselves into the local nervous system.
But here's where the story gets really interesting. The researchers identified the mesenchymal subtype.
The big discovery.
This is the hidden identity that evaded detection for decades. When scientists looked at these cells, they realized they were basically double negative.
Right, double negative.
They didn't possess the frantic division-yelling markers of the basal cells, nor did they have the wiring markers of the neural cells. So what were they doing?
Instead, their genetic profile revealed them to be acting like, almost like saboteurs.
Saboteurs, I like that.
They were actively expressing genes responsible for angiogenesis, which is the physical construction of new blood vessels.
Supplying the tumor.
Exactly. They were also undergoing something called epithelial mesenchymal transition, or EMT, fundamentally changing their shape to remodel the surrounding tissue, driven by genes like BMP7 and VIM.
It's like discovering that alongside the frontline soldiers and the communicators in an enemy camp, You have an entire hidden engineering core.
That's a perfect analogy.
They aren't actively fighting, right?
Right.
They're quietly laying down highways, building supply lines, and restructuring the terrain to support the broader invasion.
Yeah, they're the architects.
But this raises a super counterintuitive question. If you have a tumor subtype that is actively building blood vessels and remodeling your tissue, you would assume that is a highly lethal variant.
You would think so, yeah.
But the clinical survival data showed something completely different, didn't it?
It really did. The clinical data revealed that patients whose tumors were predominantly this newly discovered mesenchymal subtype actually exhibited the most optimistic survival outcomes among the three.
Wait, really? The best survival outcomes?
Yeah. The basal subtype, with its frantic, unchecked cellular division, remains by far the most dangerous and lethal form of the disease. The mesenchymal tumors, despite their complex engineering of the local environment, they simply don't divide and conquer at the same overwhelming speed.
So the hidden subtypes that we didn't even know existed is technically the one you would prefer to have if forced to choose.
Ironically, yes.
So, recap number 2 for you guys. The binary system is dead. Welcome the NBM system. Neural, basal, mesenchymal. Basal is highly proliferative and aggressive. Mesenchymal is the newly discovered architect subtype with better survival.
Right.
But here's a question for you. If you're a doctor treating a patient, how would you quickly figure out which of these 3 distinct personalities the tumor has without doing million-dollar single-cell sequencing on everyone?
Right, because that perfectly illustrates why shattering that binary illusion was so critical, but also presents a huge hurdle for patient care.
Exactly. Imagine being a doctor in a standard community hospital. It's wonderful that scientists at top-tier research institutions can use these expensive tools to map out a tumor's exact blueprint. But you don't have that technology or that budget or that computational power.
Nope, you have standard lab equipment.
So how are you supposed to use this complex genetic data to diagnose the patient sitting right in front of you?
Well, the researchers anticipated that exact bottleneck. They knew they had to translate this mountain of high-tech transcriptomic data into a practical, cheap, everyday clinical tool.
And how did they do that?
They achieved this by creating a diagnostic flowchart using a standard technique called immunohistochemistry, or IHC.
IHC, okay.
This is a really basic staining process that allows a pathologist to look for specific proteins on a glass slide using just a regular hospital microscope.
See, this is arguably my favorite part of the deep dive because it distills tens of thousands of complex genetic markers down to just 3 visual checkpoints.
It's incredibly elegant.
They looked at 30 different patient samples and built this beautifully simple algorithm for the pathologist looking through the microscope. Step 1. You stain the slide for the Ki-67 protein.
Right, Ki-67.
And Ki-67 only appears when a cell is actively preparing to divide. If 25% or more of the cells light up with this stain, you stop right there.
Boom, you have your answer.
Exactly. You were looking at the aggressive, fast-dividing basal subtype.
But if the slide remains mostly dark in step 1, you move to step 2.
Okay, step 2.
You apply a stain for a protein called chromogranin, or CHG. This is a classic marker for neuroendocrine cells.
CHG, right.
Pathologists use something called an H-score, which measures both the intensity and the percentage of the stain. If the H-score for CHG is 75 or higher, you've positively identified the brainy, nerve-wiring neural subtype.
And if you hit a double negative, if the cells aren't dividing fast and they aren't wiring nerves, you move to step 3.
The final step.
You stain for CTGF, the connective tissue growth factor, or CCN2. If that stain lights up with an H-score of 75 or higher, you have successfully exposed the hidden mesenchymal saboteurs.
That's it.
3 physical stains, 3 steps. A completely new understanding of the disease, adapted for a standard hospital lab.
And the practical application of this flowchart was brilliantly demonstrated in a specific case study from the paper involving a patient designated as ONB-311.
Oh, the ONB-311 case. Walk us through that.
So when pathologists analyzed ONB-311's tumor using these specific markers, they discovered it actually wasn't a uniform mass. It possessed a mixed personality.
A mixed personality. That sounds bad.
It is. The bulk of the tumor was the aggressive basal subtype, but scattered throughout were distinct pockets of the mesenchymal subtype.
Dealing with a mixed personality tumor sounds like an absolute nightmare for an oncologist. I mean, how do you attack an enemy that is deploying 2 completely different strategies at the exact same time?
It's historically been very difficult. But to figure this out, the researchers created a living laboratory. They developed a patient-derived xenograft, or PDX model.
A PDX model, so a mouse.
Yeah, a mouse. They took fresh living tumor tissue extracted directly from patient ONB-311 surgery and implanted it into an immunodeficient mouse.
Wow.
This allows the human tumor to grow and behave exactly as it would in the human body, giving researchers a real-time arena to test targeted therapies.
That is incredible.
Right. If we connect this to the bigger picture, for a rare cancer that historically lacked any standard chemotherapy regimen, this is the dawn of true precision medicine.
Because previously, doctors had to deploy broad-spectrum, generalized chemotherapy and simply, hope the tumor responded.
Exactly, just cross your fingers. But with the ONB-311 mouse model, they could look at the precise cellular personalities and select their weapons accordingly.
Right.
Because the tumor had a massive basal component, those rapidly dividing cells, they administered doxorubicin, which is a chemo agent specifically designed to disrupt the cell cycle and halt rapid division.
But they knew that doxorubicin wouldn't stop the hidden saboteurs.
Nope, the mesenchymal cells would ignore it.
Because the tumor also featured that mesenchymal component, the cells actively building blood highways. They simultaneously deployed a drug called endostatin.
Endostatin, right.
Which is an inhibitor of tumor angiogenesis. Its entire biological purpose is to stop the tumor from building its physical supply lines.
And the results in the living model were just striking. When they combine these highly targeted drugs with standard radiation therapy, the human tumors inside the mice shrank dramatically compared to the control groups.
Like a noticeable difference.
Oh, huge. The radiation and doxorubicin combo devastated the aggressive basal cells, while the radiation and endostatin combo specifically crippled the mesenchymal supply networks.
Let's take a second to fully appreciate what just happened here. Time for a quick recap. We moved from grinding up an unreadable tumor smoothie to isolating pure cancer cells, to finding a hidden subtype, to building a cheap hospital test with just 3 protein checks, Ki67, CHG, and CTGF, and then proving that deploying specific drugs against specific cellular personalities actually destroys the tumor in mouse models.
Yeah.
It is literally a masterclass in modern oncology.
It really is.
But it leaves a lingering fundamental question for us. We now know how to diagnose and target the tumor in its mature state. But a lingering question remains, where does this tumor actually begin its life? What pushes a normal, healthy nose cell to suddenly turn rogue?
Right, the origin story. To answer that, the researchers had to reverse engineer the tumor's development using a computational method called pseudotime analysis.
Pseudotime analysis?
Yeah, you can visualize this as mapping a massive dynamic family tree. Since they had the single-cell data from both the healthy tissue and the cancer tissue, they could lay the genetic profiles out on a timeline to find the exact fork in the road.
They were looking for the exact moment the healthy cells programming got corrupted.
Exactly. And they traced both the normal cells and the cancer cells back to a shared common ancestor, the horizontal basal cells, or HBCs.
Okay.
These are normal stem cells in the nasal cavity. And at a very specific, pivotal moment in their development, a bifurcation occurs.
A split.
Right. Down the healthy path, the stem cells mature normally into the sensory receptors that allow you to smell a cup of coffee. But down the corrupted path, the standard genetic programming just drops off.
Drops off how?
Genes responsible for maintaining normal tissue boundaries, like TWIST1 and EPCAM, they plummet, while tumor-promoting genes skyrocket. That cellular pivot point is the genesis of the cancer.
Wow. Understanding that developmental origin story, the childhood of the tumor is vital because it reveals exactly how the tumor constructs its defenses, right?
Totally.
And the researchers uncovered some incredible pieces of genetic armor in that single-cell data. They noticed that O and B cells heavily overexpress a specific protein marker known as TUBB3 or TUJ1.
Yes, TUBB3. This single protein marker fundamentally alters the clinical landscape for 2 distinct reasons.
Okay, so what does this all mean for you listening right now?
First, it serves as an incredibly bright diagnostic beacon. See, there is another aggressive cancer called sinonasal melanoma that occurs in the exact same location.
In the roof of the nose.
Right. And under a conventional microscope, using older diagnostic criteria, ONB and sinonasal melanoma are practically visually identical.
Wait, really? Identical?
They are notoriously easy to mix up. The researchers actually found a patient in their own study who had been preoperatively misdiagnosed.
Oh my gosh.
But when you apply a stain for that TUBB3 protein, O and B cells light up intensely while the melanoma cells remain completely dark.
Wow. So a simple protein stain can instantly prevent a devastating medical mix-up. But as you dig into the literature on TUBB3, you realize it has a very dark secondary function, right?
Yes, it does.
The tumor isn't expressing this protein by accident. In the world of oncology, TUBB3 is a heavily documented marker for physical resistance to vincristine, which is a very common, potent chemotherapy drug.
Very common.
By expressing this specific protein, the tumor is actively showing us its physical armor. It is essentially broadcasting to the oncologist, do not bother deploying vincristine against me. I already have the biological shields in place to deflect it.
Exactly. The single-cell resolution allowed researchers to read the enemy's entire defensive playbook before a single drug was even prescribed.
Which is incredible.
And it wasn't just vincristine. They also detected high expression of a gene called ERCC1, which in lung cancers is a known shield against the chemotherapy drug cisplatin.
Another shield.
Right. Additionally, they found elevated levels of NME2, which correlates directly with resistance to 5-FU, so we are no longer guessing which drugs might fail. The tumor's individual cells are mathematically warning us.
It is the biological equivalent of intercepting the enemy's encrypted radio transmissions.
It really is.
And to add one final incredible layer to this ecosystem, we really have to talk about the tumor microenvironment, or the TME. Because the tumor isn't just an isolated ball of rogue cells. It is an active manipulator of your body's own immune system.
Oh, absolutely. When the researchers analyzed the TME, they noticed a highly specific communication network originating from our new friends, the mesenchymal cells.
The saboteurs.
Right. These saboteurs are incredibly social. They're constantly sending chemical signals to the surrounding normal tissue, particularly to a type of immune cell called a macrophage.
Okay, macrophages. In a healthy body, macrophages act as, like, the local police force, right?
Yeah, they're supposed to identify invaders, swallow cellular debris, and basically maintain order.
But in this specific tumor environment,
the single-cell data revealed a massive abundance of immunosuppressive tumor-associated macrophages.
wow. So the cancer has essentially bribed the local immune police to look the other way, and in some cases, actively protect the tumor from the body's other defenses.
But bribe the police. That's exactly what's happening.
But identifying that corruption opens up a completely new frontier for treatment, doesn't it? If the tumor is fundamentally relying on these hijacked macrophages to survive, future immunotherapies don't even need to attack the cancer directly.
Right.
If a drug can specifically target and reprogram those corrupted macrophages, you could strip the tumor of its protective detail and just let the body's natural immune system do the rest.
It's the ultimate promise of this technology. We're moving beyond blunt force trauma to the body, toward dismantling the tumor's communication and supply networks from the inside out.
Okay, let's synthesize exactly what this journey represents. We started with a rare, poorly understood medical mystery located at the base of the brain, with a 50-50 survival rate.
Right.
For decades, the medical establishment confidently treated it as a straightforward binary disease. But by utilizing single-cell RNA sequencing to map over 96,000 individual cells, scientists unblended the tissue smoothie.
They unblended it.
They isolated the chromosomal chaos, uncovered a hidden third identity operating as a biological saboteur, tracked its exact evolutionary origin from a normal stem cell, and mapped its physical armor against specific chemotherapy drugs.
And most importantly, they translated all of that bleeding-edge data into a simple 3-step visual test that any hospital pathologist can use tomorrow, proving in living models that matching the drug to the specific cellular personality actually destroys the tumor.
Exactly, which matters to you whether you're an oncologist prepping for a tumor board or just someone fascinated by biology. It proves that zooming in to the fundamental individual unit of life can clear away decades of medical fog.
It really does, which leaves us with a pretty profound structural realization about modern medicine.
Yeah?
This study proved that a disease we confidently thought was binary, just A or B, was actually hiding a completely different option C that changes absolutely everything about how we diagnose, understand, and treat it.
Option C.
Right. So as we begin to apply this level of single-cell sequencing to the rest of the human body, it raises an unavoidable question. What other deeply established binary diseases are secretly hiding a third, fourth, or fifth biological identity just waiting to be discovered?
Man, it makes you look at every single medical textbook currently sitting on a shelf just a little bit differently, doesn't it? We'll leave you to think about that.